Okay, so this lecture is about mitosis. So mitosis is the process by which a cell replicates, it's the process by which a cell separates after all of the genetic information and pieces inside of the cell have been doubled, and then the cell separates into two identical copies of itself. So that's what we're talking about today. So cell reproduction is, we're going to use a few different models to look at and to see this overall process. But the basic principle is that a single cell divides to form two cells. And then these two cells are identical to the original cell. So this is talking about a sea urchin that begins as a single cell, and that single cell divides to form two cells, and after four rounds of cell division, there's 16 cells, and then after many rounds of cell division and growth, the individual develops into a complex multicellular sea urchin. So the original single cell was the ancestor of every other cell in that organism. And then once the mature organism is fully grown, cell reproduction is necessary to repair and regenerate tissues, like we're looking at this starfish here. So organisms use cell reproduction, for growth and maintenance to repair cells and tissues. So the process that we're going to be talking about is mitosis. And during mitosis, there's a process where all of the genetic information in the nucleus of the cell replicates. So now we have two copies of all of the genetic information, so we can split into two cells. When we're talking about genetic information, all of the complete genetic information in an organism in each cell is called its genome. So there's a few different words that we use when we're talking about genetic information. We talk about chromosomes, genomes, karyotype. All of this is just basically the DNA of the cell, which holds all of the genetic information for that organism. So the region of the cell containing the genome is called the nucleoid, so that in a eukaryotic organism is found in the nucleus. So what we're looking at here with these 23 pairs of chromosomes, this is called a karyotype. And you can see that there's two chromosomes for each of these, 1 through 22, and then you see the 23rd chromosome in mammals, in humans. This is the sex chromosome. So you either have an X or a Y. And then when those two come together later, we'll talk about that. XX is female, XY is male. Okay, so just a little bit of background information before we get into the actual process of mitosis. So when we're talking about chromosomes, they can exist in two ways. You can have one chromosome, which is unduplicated. Usually it's called one chromatid. Or you can have two chromosomes, which is an identical copy of each chromosome. Those two chromosomes together are called sister chromatids. So when we talk about genetics and nucleic information, we usually use a female gender, like sisters or daughters. Has nothing to do with being male or female. It's just how we talk about things that are the same. So these two sister chromatids are the same. We could also call them brother chromatids. That's just not the nomenclature or the name that we use in genetics and biology. So those two sister chromatids just mean they're identical copies of each other. When we talk about daughters later, like a daughter generation, would just mean it's the next younger generation. Again, it has nothing to do with being male or female. Okay, so in eukaryotic organisms, the genome comprises of several double-stranded linear DNA molecules. So it's important to indicate that these are linear because there are some organisms that have circular DNA. We're not going to talk about any of that today, but bacteria, for example, can have circular DNA. What we're talking about is linear DNA molecules. So each linear DNA strand is called a chromosome. A cell contains 2 matched chromosomes, sister chromatids, and we call that diploid. So again, di means 2, so that means we have 2 copies. The 1 chromosome here we would call this haploid, which is half of the total genetic information. So the matched pairs of chromosomes are called a homologous chromosome. So homo means same. So these are 2 of the same chromosomes. They're the same length and they have the same specific segments of DNA. Those are called genes. So what the definition of a gene is it's a nucleotide segment. So it's DNA that is going to code, it's going to have the information for a protein. So we have genes that code for proteins, and we'll talk about this a little bit later when we talk about DNA and protein synthesis. But all of this is happening in the nucleus of the cell. So the karyotype, which we saw a picture of before, is the number of chromosomes, and it's a picture of the number of chromosomes. And different organisms will have a different number of chromosomes. The number of chromosomes is not related to the complexity of the organism. So for example, a rabbit has 22 pairs of chromosomes, and so does a coffee bean. So the number of chromosomes, it does not mean the more chromosomes you have, the more complicated the organism is. Here's another picture. These are the karyotypes of a human versus a chimpanzee. Chimpanzee has 48 total chromosomes. We have 46 or 23 pairs of chromosomes. And then here's a picture of a fern just to illustrate that, you know, the number of chromosomes does not have anything to do with complexity. This fern has 1,260 chromosomes. So obviously way more chromosomes, but a much less complex organism. Okay, so human chromosomes are linked together by a point in the middle called a centromere. And the centromere is going to be important during mitosis. This is where the 2 sister chromatids are being held together. So this is a chromosome here. This is a homologous chromosome with 2 sister chromatids. And then this part in the middle right here would be called a centromere. So this lecture is about mitosis. Mitosis is one part of what is called cell cycle. So cell cycle is involved in cell growth and cell division. It's where one cell grows and divides and splits into 2 identical daughter cells. So those 2 daughter cells are exactly identical to the original parent cell. The cell cycle has 4 phases. It has G1, which G stands for growth. So growth 1, if the cell is ready to go through cell cycle, it'll go from G1 to S. If the cell is not ready to replicate and go all the way through cell cycle, it will go into what's called G0 or gap 0. Sometimes this is called senescence, kind of like the cell is sleeping, like it doesn't want to grow and divide and replicate. But we're just going to pretend we're replicating the cell, so we're going all the way through cell cycle. So from G1, this is a growth phase where all of the stuff in the cell is replicating. So like mitochondria, or if it's a plant, chloroplasts, or the Golgi apparatus or ribosomes, or any of the things that you saw in that picture of a cell, all of those things are replicating. Then we have the S phase. The S phase we're going to talk about a little bit more when we talk about DNA replication. The S phase, S stands for synthesis, and this is when all of the DNA is replicating. It's synthesizing DNA. So however much DNA there is in that cell, it's going to double it. So in the S phase. So then we're going to go into G2 phase. So this is the second growth phase where everything else in the cell is replicating. And then we're going to go into mitosis. So actually G1, S, and G2, all of that can also be referred to as what's called interphase. Other than interphase, the other phase is called mitosis or M phase. And that's what we're going to focus on. When we get to the beginning of M phase or mitosis, everything in the cell has been duplicated. So what happens in M phase is now we have this cell that has twice as much information in it that we need, and we're going to separate that cell into two identical cells. So that's what's happening now. We've gone through G1S and G2. Now we're ready to split the cell into two parts. So this is a little more information about what I just explained about G1S and G2. And once we are ready to go into M, mitosis, we're going to split all of this apart. So G1, we are replicating everything in the cell. S, we're doubling the DNA. And then G2, we're making all of the other organelles and getting ready to divide. Okay, so mitosis is a multi-step process. Some books break it down into 4 steps. Your book breaks it down into 5 steps. So everything in the cell is being aligned and separated, and it is going to result in 2 identical daughter cells. So there's 2 major portions. The first portion is to accomplish nuclear division. So nuclear division is when all of the DNA is being separated perfectly and identically into 2 new cells. And then the second portion accomplishes cell division, where it separates these 2 cells. So here's mitosis. So it goes from prophase to prometaphase, prometaphase to metaphase, metaphase to anaphase, anaphase to telophase. So when we look at this in lab, we're going to see cells that are in all different stages of mitosis, and this would be a good slide to reference back to for you to be able to identify what cells are in what phase of mitosis. So the first portion is prophase, prometaphase, metaphase, anaphase, telophase. The second portion is the cytokinesis, where the cell actually separates and has 2 new cell membranes, and now we have 2 new cells. Okay, so let's go through each of these in a little more detail. So first is prophase. So what happens in prophase is the chromosomes condense and become visible. The nuclear envelope, which is the nuclear membrane around the nucleus, starts to break up. Then the centrosomes, which are organelles, begin to move to the opposite poles of the cell. So you can think about it as kind of like the north and south pole of the Earth. And then microtubules emerge from the centrosomes. So microtubules are actually part of the cell's cytoskeleton, and they are going to help separate everything inside of the cell. The next phase is called prometaphase. The chromosomes continue to condense, and the rest of the nuclear membrane disappears. Centrosomes are on opposite poles of the cell, and the microtubules will extend from the centrosomes, and each sister chromatid attaches to the microtubules. So from prometaphase to metaphase, the chromosomes are lined up. So if we were using kind of like an Earth, like a globe analogy, you would see these centrosomes are on like the north and south pole, and the chromosomes are going to like line up on the equator. During anaphase, the centromeres are at the poles. The centromeres are, the microtubules are attached to the centromeres. And then the sister chromosomes, chromatids, are pulled towards opposite poles, and the cells begin to elongate. So this is twice the amount of genetic information that one cell needs, is what we have here in metaphase. What we're doing in anaphase is now this is going to be each of these is going to be all of the genetic information that we need for these 2 cells. So this is going to pinch off in the middle and become 2 cells. In telophase, the chromosomes arrive to opposite poles and begin to decondense, and then nuclear material surrounds each set of chromosomes. The nucleus is going to start reforming. Now we're done with mitosis, and we're done with the first portion of mitosis, and the second portion is the cytokinesis. So we've replicated everything in the cell, we've separated it now into 2 complete sets of information. Now we're going to go from one big double cell to pinching off in the middle, this cytokinesis, and having two identical cells. So a fissure is formed called a cleavage furrow, which starts to separate these two cells, eventually until the cell is divided into. This is a good picture of this showing the mitosis being completed and this cleavage furrow, this contractile ring, kind of starting to separate these two cells and then ultimately ending up with two complete new daughter cells. This is something that we would talk about more in a different class, but cancer is when you have mitosis that is not being regulated. So we didn't talk about that at all in this class, but this process of mitosis is very highly regulated because it is energetically expensive, and we only want cells growing and dividing when it is an appropriate time. Cancer is an example of unregulated cell growth where these cells are growing and replicating out of control. And a lot of these checks and kind of stop signs along the way of cell cycle that prevent cells from growing when they shouldn't they've been overridden by this cancer. So cancer is when mitosis is in overdrive and these cancerous cells are growing and dividing, growing and dividing, growing and dividing, and producing some sort of cancerous growth or tumor. So again, we'll look at all of this in more detail in our lab. I'm going to stop here for mitosis.